Welcome to biology. In this video, we are going to briefly introduce the concept of genetics. Now, genetics is actually the study of heredity, and you need to know that definition. Now, Gregor Mendel is considered the father of genetics, and he was a monk in the early 1800s, and he studied heredity by using pea plants. And heredity is actually the set of characteristics that an individual receives from their parents. So genetics is the study of this characteristics that we're getting from parents. Now, Mendel did some interesting, very straightforward experiments, and he used pea plants. And I have this diagram here on the side. You can see that he started with these things called true breeding parents, which means that those purple flowers were always only producing purple flowers every time that they made offspring, and those white flowers were always only producing white flowers whenever they were crossed with another white plant. So that's how he knew they were true breeding. And Mendel discovered that the offspring of a genetic cross are going to receive one set of genetic information from each parent. So one set from the mom, one set from the dad. But here's the thing. He discovered that these traits that the offspring receive are not actually a blend from their parents. So he thought that if I take a purple flower and I cross it with a white flower, that will produce a light purple or an in-between color. But that wasn't the case. What he found was in his F1 generation, or that first set of offspring from the parent generation, those all were purple. But then he asked himself a few more questions, and then he crossed those F1 generation individuals with each other, so a little bit of plant incest going on here, and he crossed them and he got the F2 generation. Now, in the F2 generation, he found that 3/4 of the offspring were all purple and 1/4 of the offspring were all white. As we go through this unit, you will figure out how we are able to predict the possible offspring of a genetic cross using this same method and these same calculations that Mendel came up with. Some of the vocab that you need to know in this unit include the terms gene and allele. These two terms students often get mixed up, so take a moment and make these into flashcards to practice with. A gene is an inherited unit or a section of DNA that determines a specific trait. Now, an allele is just a specific version of that gene. So say we have a gene that codes for the height of a plant. There can be two different versions of that gene. So there could be the allele or the version of the gene that creates a tall plant, or there could be the allele or version of the gene for a short plant. Now, in Mendel's experiment, the purple flowers have a different allele from the white flowers. So these alleles are two different versions of the DNA that codes for flower color. Another set of vocab terms you need to know are genotype and phenotype. Genotype is the genetic makeup of an organism. So what alleles or what genes are present there? So genotype discusses the genes. Typically, the genotype is represented with two letters. So it might be two capital letters, two lowercase letters, or one capital and one lowercase letter. Remember, Mendel figured out that you get one copy of DNA from each parent. So you get one set of characteristics from mom, one set from dad. That's why there's two letters here. One represents what you got from mom, the other represents what you got from dad. The term phenotype, these are the physical characteristics of an organism. So what does that organism physically look like? So this would be a description. Is it tall? Is it short? Is it purple? Is it white? You also need to know the vocab terms homozygous and heterozygous. So homozygous is when both alleles in an organism are the same. So is it capital A, or is it lowercase a? So homozygous, we are referring to that genotype. Heterozygous is when those two alleles are different. So we have capital A, lowercase a. Again, we are referring to the genotype. Now, in Mendels Experiment, the true breeding flowers are homozygous, which means both of their alleles are the same. So both are purple or both are white. Now, the hybrid flowers are heterozygous, which means they have one copy of the purple and one copy of the white allele. Again, remember, we've used these prefixes, homo and hetero, throughout the course. Every single time we've used the prefix homo, we're talking about something that is the same. And every time we've used the prefix hetero, we're talking about something that is different. We also have the terms dominant and recessive. Dominant refers to the form of a gene that is expressed anytime it is present. So it rules or dominates or takes over that recessive form. And this is represented with a capital letter. Now, recessive is much shyer. It does not express itself if that dominant form is available. So this is repressed or hidden by the dominant form of that gene, and we represent it with a lowercase letter. Now, in Mendels Experiment, the purple flower color is dominant, and we know this because all of the offspring in the F1 generation that have one copy of the dominant and one copy of the recessive, so they are heterozygous, all of those were purple, because that purple was the dominant color and it took over and expressed its phenotype. So we have our trait pairings. We have homozygous dominant, homozygous recessive, and heterozygous. And we'll go through each of these one at a time. Homozygous dominant is what happens when both copies of the allele are dominant. So we have two capital letters. We have a dominant phenotype being expressed. Homozygous recessive is when both copies of that gene are lowercase, or both copies are recessive. This is the only time that the recessive phenotype can actually be expressed. The third type, heterozygous. This is when one allele is dominant, the other allele is recessive. So we have one capital and one lowercase letter. Because there is at least one capital letter, the dominant phenotype is going to be expressed. Now, when we write the heterozygous genotypes, we always write the capital letter first. And it makes sense because it's the dominant allele. It wants to take over. It wants to be first. It wants to be large and in charge. So if we were using the letter B to represent our allele, capital B, lowercase b would be the correct way of writing heterozygous. Lowercase b, capital B would not be correct because we don't see them in that order. I put all of this information into the chart here, so the difference between homozygous dominant, homozygous recessive, and heterozygous. This is a good way to compare these traits. If you are getting them mixed up, I encourage you to pause the video here and jot this down into your notes. Now I want us to do a little review. I would like you to match each of these 5 terms with their possible example. In summary, you should be able to define each of these terms. You should be able to explain the difference between genetics and heredity, the difference between a gene and an allele, the difference between genotype and phenotype, the difference between heterozygous and homozygous, and the difference between dominant and recessive. Please leave your questions here at the end. I know this was a lot of information and we went quickly through it. You can always re-watch these videos, and I hope that as you watch this video, you were able to learn something.